Hi, student. Today you will learning from Thai teacher. My name is Atchaneey Chamnansinp. Today I will bring you go to the new world. It's about diatom. But before I will do that, I have some question to ask you. Do you know the fish? I mean, animal in the water. Why? What's about the food of them? Do you know?
The fish, what they eat.
Yeah, what them eat. You try to understand because my English is not that, you know. Yeah.
Some eat algae.
Algae.
Yes.
Okay. And another? Plankton. Do you know plankton? Oh, great. And what about you?
Yeah, that's pretty much all I know. Yeah.
Phytoplankton.
Phytoplankton. Okay.
Other fish.
Okay.
Yeah.
Okay. And what about you?
I would say.
Okay. Okay. Let's see. Today, I will tell about that later. And today, I want you to know about the background of diatom and identify some species of diatom. It's look hard work, but it's not. I hope so. I will start from plankton. Plankton is have two different. It have zooplankton and phytoplankton. Zoo does mean is animal, and phyto does mean they can photosynthesis. Yeah, it have two kinds. And diatom is a phytoplankton. Now, can you imagine? Yeah. Have anyone no idea about plankton before? Can you? Okay.
It's very small, isn't it?
Okay.
Very small.
Okay, very small.
Yeah.
But I will tell you, it's very important. Yeah. To see, it is zooplankton. You see that before?
Yeah.
Yeah. It is the larvae of octopus.
Okay.
Yeah. It's very cute.
Yeah.
And it is the larvae of shrimp. Yeah. This one is fish. And that is what you tell me that's some fish. You can see something. It look like then became too adult. You can see shell.
Is it sort of a lobster or something?
Lobster.
Crabfish or?
No.
No.
It is a shell. I'm sorry.
Oh, okay.
Yeah.
Yeah.
You can see something they produce super.
Oh, yes.
Yeah. And it is a crab. And another one, maybe you know, is octopus. Coffee pot. Yeah.
Yeah.
You use them to taste something toxic or something. And another one, you see, this means zooplankton is all of them is animal. They cannot photosynthesis and it looks small, but you can see by your eye. And some of them you cannot see. You just see under the right microscope. Yeah, I let you see this picture. Do you know this one?
Jellyfish.
Yeah, a giant jellyfish. You see, this is kind of zooplankton too, because
Is it plankton?
yeah, it's plankton. Jellyfish is plankton because plankton, that means them cannot control to swimming. When have a wave in the sea, they just to go for a wave. When the wave go, they go that way. And this you see, some people just to take them go to somewhere. They cannot do anything. You see.
Okay.
Yeah. I think human is dangerous more than shark.
Yeah.
Then bring to eat in Japan. Yeah, it's very sad. And you see it is a phytoplankton. Phytoplankton, you can see them have chloroplasts for photosynthesis. You can see green and some of them is blue. Yeah, because it have many kind of pigment to use photosynthesis. And all of them is a diatom. It is diatom, it is diatom, diatom, and it is green algae. It is diatom, diatom, and it is dinoflagellate. You hear about dinoflagellate before? Yeah, you just know because because dinoflagellate them can produce the toxic. You hear some person to sick when you eat the shellfish.
Mm-hmm.
Yeah, some of them is dead in Canada because from some toxic from microalgae to keep in mussel, and when they eat mussel and then just to dead in about 24 hours. Yeah, it is many toxic in that. But diatom is a little bit have toxic, but it's dangerous too. Yeah, it's not bad. When you go to the ocean and you see the water of the ocean have some color, it's not, maybe you cannot see that because it have some area. That means have a plankton boom. Yeah, it is a plankton boom. Maybe we said it's very quickly to grow, and that make the water is not enough oxygen in the water, and animal became to death. Yeah, that's it's not good. That means the nutrient from the land is full very much and make them go very well, and it's not good. Yeah, but it's difficult to take care about that because it depends on the current of water to make the nutrient that area so much. Yeah, but I used to tell the important thing, if that area have a news in the newspaper or something to say, oh, it's have plankton boom in your area, you just to take care, don't eat food from the sea that time.
Okay.
Yeah, because maybe it's have toxic or something like that.
That's a good lesson.
Yeah, good to know.
Yeah.
Oh, it's disappear something. And this, I will show you. Yeah, in the food chain, it's very important thing. It is phytoplankton start here, the first primary producer, and zooplankton to eat them, and fish larva and small fish and predator, the big fish, and yeah, the final is people. And yeah, it is, I want you to know, it is some zooplankton too, is very important too. We call it kill. Yeah, you know that? This is the important food of whale. Yeah, yeah. There is so much. Yeah. I will tell you, if it's not have phytoplankton, it's not have zooplankton, it's not have food shell, it's stop because it's not have food in the sea. That's mean why it's important. Yeah. And we will go to diatom. I think so. Oh, it is a little bit I want to tell about the size. We call separate. I mean, we call a different name about plankton from the size. Yeah, you can see from that it is 0.2 to 2 micrometer. It's we call picoplankton, and nanoplankton is about 2 to 20 micrometer, and microplankton is 20 to 200, and mesoplankton is 200 to 2 millimeter, and macroplankton is more than 2 millimeter. Yeah, and grow is very big, more than another thing, more than 1 centimeter. You can see by your eye. If it's start more than 2 millimeter, you can see. Yeah, you can see. But all of them, when we study, we use light microscope and some microscope to study. This I will show you about how to sampling. We use that one, we call plankton net. Yeah. It is the important instrument when you study about plankton. Yeah. It has many size of them. I mean, what you call that? I'm not sure.
The maze of the net.
Yeah,
Maze
that's
size.
true. Yeah, maze size. Many depend on what you want to study. And it's really very expensive. Yeah. Sometimes we go by boat and we lost them.
Okay.
Yeah.
Uh-oh.
Because when we put out the boat and the boat go and sometimes the water current.
You took it.
Yeah, we cannot.
Not good for the
find.
PhD budget.
Yeah, it's very expensive. Yeah, we go back to diatom. Before I want to tell you what's the diatom. I mean, the diatom is very important because in phytoplankton, about 80% is the diatom. Yeah, that means most of phytoplankton is diatom. Can photosynthesis and give oxygen for the world and keep carbon dioxide and the first primary production. Yeah, it's very important. Yeah, and very fantastic. Most people here don't know about diatom. No. It have something wrong here. That's okay. Yeah, all of them. This is instrument we use to study light microscope, and that one is scanning electron microscope. Because some structure we see from this one, and some structure we cannot find, we just to find from that one. But we just to use both of them to study. You cannot use that, or you cannot use that only because, yeah. Oh, sorry. But some species you can use only this if you have a reference to refer about the picture. Because some diatom is very big cell. It's okay to use this one. Yeah. But scanning is very expensive to use, but now it's very important. And this I will show about the picture. Scanning electron microscope to compare about LM. It is you see from light microscope. When you see from here, you cannot see some process. It looks like this one and this one. And the good detail of this you cannot find from that. That's why you want to scan electron microscope to study that. I will pass this quickly because it's only for tell you know about to use light microscope and electron microscope. These two. This is light microscope. You can see something here, some process here. But when you pick this cell to see by SCM, you cannot see. You see? You cannot see. Yeah, that's why you use light microscope to study too. It is a good detail too, but you cannot see this process. Yeah, you just use both of them. And this is not enough. You just use these two because you see another detail of them. What do you think is beautiful or not?
Mm-hmm.
Yeah. You agree with me that dome is very beautiful.
Yeah.
Yeah. I'm excited every time when I pick cells to see by scanning. Yeah, I want to see what is that. It's very fantastic. All of them is my picture. Yeah, these two to compare. Some structure you see. It is, yeah, you cannot see it from that one. Most of them, the picture is the same cell. When I take a picture from LM and I pick cell to take a picture with scanning to compare what's different. And now I want you to know about structure of diatom. You can imagine about box. Did you know? Yeah, box and have a lid.
Lid.
Yeah, a lid to cover. That means it look like that. It have a cover here. That means it's big more than this one. And all of them here we call fastu. This is a cell of diatom and have light material inside. Fastu that's produced from silica. Yeah, it is a silica. That make our diatom is very easy to study because it's most of them is a robot. It's not delicate. Yeah, but okay, some of them is delicate, but if they produce silica, it's a hard cell. It's different from dinoflagellate or another macroalgae because they not have this one. Yeah. And before we go to identify diatom, it's important you know about some structure of them. Some diatom is a large cell, and some is look like this. And you can see some diatom is not have something look like this. Yeah, it's not have this one. That we use to separate diatom to two groups. It's a centric diatom and dendritic. I'm sorry, centric diatom and pennate diatom. Yeah, but I don't want you to know so much because this is the first class. Maybe the next I will tell you. Yeah, it's easy to know today. Now you can imagine about the cell of diatom? Yeah. And sometimes them call, that you see maybe you have something to check. Oh no. But it has something I want to tell you to know because I feel it's interesting. It is we call, maybe we go back this one before. Okay. Yeah. We call this one is epival, and this is hypoval. Sometimes you can say it is the oldest valve, and it is younger. Why that? Do you know? Do you have any idea? Why it is the old and it is the young? But it's the same cell here. It's only single cell, one cell, but it have two valves. Do you have idea? Why you all with more than your younger sister? Why? Okay. That's because when you see when them divide cell, then we will separate the valve, separate to look like this. And this one go here, small one. And the low the hypoval, then will be produce a new one here.
Okay.
Yeah, and this produce a new one here. That means it became to a perfect cell. That means it is old and it is a young and when diatom divide again and again and again, they became to a small one. It look like this. Yeah, and it is then became to a sexual reproduction again, because then can divide. It look like this and sexual reproduction to produce sperm and mix with egg and swimming. When you see sperm of diatom, maybe yeah, some person don't believe that it's plant. Yeah, but it's plant. Then have chloropart to photosynthesis. Okay. And this is the important thing to use to identify species of diatom. I will tell you only 2 names for today. It's not hard word. It is we call lanceolate, you see? Yeah. It look like this. Yeah. It look like this, we call lanceolate. And it is look like this one. This and this we call linear. Yeah. You should to remember because I have work for you to do. Okay. And it is important thing to study. Study is the law of poroid, and interstudy is a bank of the bank area. That means if it is a cell of diatom and then have a poroid to sit here, a small poroid, and when you see look like this, it look like have step here. Maybe it's hard to see from this. Maybe you write down somewhere. We call it look like structure. It look like a hole, a small hole. Maybe them have a compact hole. It's many hole, but some of them is only one. And when them stay together, look like this. We call this style. And it look like this when go to the cell. Can you imagine? Yeah, it look like this. And we count the number of this in 10 micron. We use it, no, not 10, in 1 micron to identify diatom. We just to count the poroid in 1 micron and how many of them, and we count how many study in 10 micron. Yeah, you can count it, and you can count. Can you understand? Okay, let's see if you wrong. If you wrong answer, that means you not understand. Okay. And it have something too important to know. Raphy. I tell you the first, but yeah. We call this one as Raphy. Raphy is go on in the cell, inside of the cell. When you see inside, it look like this one. It's the same, yeah. This is the outside and the inside. And Raphy then cannot join together. Yeah, have something here to separate Raphy. We call this one is central nodule. Central nodule, yeah, it's important too. And this I want to tell you to know how to measure the cell of diatom. It's the important thing for you to identify species. we call this apical axis, this one and this one, per one y-axis and this one times the axis. I have some question. What is this?
Height.
What is
Height. This is the vertical axis height.
Yeah, but this or this?
This is the per
Per one y?
yeah.
Okay, you correct. Great. Yeah, now you know. And anybody get about that or not? If this one is round, that means the diameter is the same here and here, because apical axis this one and classical axis this one, and this one is per one y-axis. Yeah, but if cell is look like this, it's half dimension. This maybe you can tell its length TH. It's correct?
Yeah.
Yeah, you can call this one. And this one you can call Y. Which? It's okay?
Yeah.
Yeah. Now you know, and maybe we can start.
Well, actually, Atchaneey, you're actually out of time now. You have
Oh,
been talking for 27 minutes now.
27?
Yeah.
Okay.
So maybe you could just tell us about what the task is about.
Okay,
And
why
then-
is very quickly? Thank you. Okay,
So
sorry.
if you tell us about what this task, the questions is
Okay.
about, and then
Okay.
I think we have to stop.
Okay. We don't have time to do the question.
I don't think so.
No,
No.
but you can just give us
You
the
can give
paper.
it,
Okay.
explain
Yeah,
what
and then
the
yeah,
idea
yeah,
is.
yeah.
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 How we can see this.
Okay, a
Yeah.
few minutes.
Explain the idea.
Yeah.
Yeah.
Sure.
Oh, I'm sorry. It's very many things to talk. If you see that one, I just it's only to measure the cell, how much to which and how much to long, and how many poroi in this in one micrometer, and how many style in 10 micrometer, and you will know about